Welcome to ICU Primary Prepcast.
Hi, I'm Maddie.
Hi, I'm Swapnil, and welcome back to another snippet for this week. So, Maddie, this week we are going to dive into hematology question. So, can you please describe the structure and function of adult hemoglobin?
Sure. So thinking first about the structure, hemoglobin is a tetramer which consists of 4 polypeptide chains. Each of these chains has its own heme group. So adult hemoglobin or hemoglobin A consists of 2 alpha and 2 beta polypeptide chains, which are all similar in size and structure. We know that the alpha polypeptide has a bit more amino acid residue, so they have 141 compared to beta, which has 146. So these 2 alpha beta subunits are arranged tightly together in a tetrahedral form that is held together by hydrophobic bonds. And the alpha and beta chains are each comprised of 7 or 8 helices named A to H that are joined by non-helical segments. In these helices, the E and F helices form a pocket for heme to bind via histidine residues. The heme component is comprised of a ferrous ion, which is held in the middle of a porphyrin ring called protoporphyrin IX. And this heme is the site of reversible oxygen attachment. Therefore, each hemoglobin molecule can carry up to 4 oxygen molecules. In terms of the function, hemoglobin has 4 main functions. So these are oxygen carriage to the tissues, carbon dioxide carriage, acting as a buffer, and the transport and metabolism of nitric oxide. So oxygen carriage is probably the main role that we all know of for hemoglobin. Hemoglobin increases the oxygen carrying capacity of blood by greater than 50 times. And the binding of oxygen to one heme group causes the ferrous ion to be pulled closely towards the protoporphyrin ring. And this slightly changes its shape. And this alters the quaternary structure of the hemoglobin molecule and thus increases the oxygen binding affinity of the other globin chains. So this is known as positive cooperativity. So basically, when one oxygen molecule binds, it makes it easier for further oxygen molecules to bind. We know that hemoglobin exists in 2 main forms. So when it's deoxygenated, it exists in the T or the 10th state, and this is not favorable for oxygen binding. But as oxygen binds, it makes it easier for further oxygen molecules to bind, and then it changes its shape to R or relaxed state, and this has a higher affinity for oxygen binding. So when we're in the lungs where the oxygen pressure is high, it allows for hemoglobin to be fully saturated with oxygen more easily, and then conversely for hemoglobin to easily release oxygen at tissues where the oxygen pressure is low. Coming then to the next role, which is carbon dioxide carriage. Hemoglobin carries carbon dioxide as carbaminohemoglobin. It has a higher affinity for carbon dioxide when it's deoxygenated and a lower affinity when it's oxygenated, so therefore releasing CO2. And this helps to aid in its role in oxygen and carbon dioxide transport. Next, hemoglobin as a buffer. So a buffer is any solution which contains a weak acid at its conjugate base, and this resists pH changes when exposed to a stronger acid or base. So in the body, the buffer binds like any acid ions reversibly to minimize the change in pH. Hemoglobin has multiple histidine residues with imidazole side chains. Imidazole has a pKa of 6.8 and can bind hydrogen ions. And hemoglobin accounts for 50 to 60% of the buffering capacity of blood. It's a weak acid and it buffers hydrogen ions and forms bicarb within the hemoglobin molecule. Intracellular bicarb increases, leading to diffusion down the concentration gradient out of cells. So hemoglobin acts basically as a very important buffer within the blood. And finally, coming to the transport of nitric oxide. So nitric oxide binds with high affinity to the ferrous ion of heme. If the hemoglobin is in its deoxygenated state, this forms nitrosyl hemoglobin. Affinity for nitric oxide is greater than for either oxygen or for carbon monoxide. However, if hemoglobin is oxygenated, the nitric oxide can displace the oxygen and oxidizes ferric ions and forms methemoglobin and a nitric ion. This reaction occurs very rapidly and can quickly sequester nitric oxide, which is a potent vasodilator. But this reaction is limited by the hydrophobic membrane of hemoglobin. Nitric oxide can also bind to cysteine residues in the beta globin chains, which form something called S-nitrous hemoglobin or SNO-HB. This reaction occurs faster with oxygenated hemoglobin, and therefore helps to regulate low blood flow as the release of this SNO HB is increased in low oxygen environments and causes vasodilation. So thought to help auto-regulate parts of the body in terms of blood flow, depending on the amount of oxygen present.
Thanks, Maddie. Again, this is another bread and butter question for the primary, and it's very important to write this in more depth. One of the common mistakes that happens during the written exam is often candidate fail to write the content in more depth and expected at this level. Also drawing the structure of hemoglobin with different porphyrin ring and central iron molecule is not a bad idea to supplement this answer. So thanks again for providing this comprehensive answer. We'll be back in week's time with another primary snippet. Till then, goodbye and have a nice time.
Thanks for listening and see you next time.